Our speaker tonight is Rob Stadler. Dr. Stadler is a scientist and a committed Christian. By God's grace, he earned a PhD in medical engineering from Harvard and MIT. He has obtained more than 180 U.S. patents and contributed to a variety of cardiac devices that have been implanted in millions of patients. Rob is passionate about resolving the apparent conflict between science and faith, a topic that he addresses in his first book, The Scientific Approach to Evolution, and in his recent co-authored book, The Stairway to Life. In a society where science is increasingly portrayed as the ultimate authority that contradicts God's word, this message is urgently needed. Please join me in welcoming Rob Stadler.
So welcome everybody. Thanks for coming, and thanks for the opportunity to speak here tonight. I'm looking forward to this. Yeah, here's the book that you can get on Amazon. But the focus of tonight is going to be really on how long-term evolution experiments. So these are experiments that are kind of provoking life to see what evolution can accomplish. And I'm going to summarize several of them and show how they show us that evolution is actually a highly constrained process. So a little teaser there. But in the big picture, what we're trying to do today is to compare two hypotheses. One hypothesis is the tree of life. So these are hypotheses for explaining how we have modern life that we have today. So you're all familiar with this. So we have a single-celled organism that through the process of evolution over millions, maybe billion years, has given us the great variety of life we see today. The alternate hypothesis, or one alternate hypothesis, is more of what I think the Bible is telling us, which is more of a forest of life, where we have different kinds of life, as explained in Genesis chapter 1. I think 10 or 11 times you see the word kinds, as you see here. And I believe that's referring to separate trees, one for each kind of life, wherein each tree is allowed to diversify quite a bit, but you're still constrained to be within that tree. So maybe one of the more famous examples is the canine, wherein wolves have diversified in lots of ways, but becoming all the breeds of dogs we know today. These figures are obviously oversimplified, but it's here for more of a cartoon example. But I also wanted to emphasize that although I'm showing these different kinds all starting simultaneously, that's not the point. That's not what I'm focused on. You can stretch these over whatever time you want. The point is that they're separate trees as opposed to one simple tree with everything combined. So if one thing to pay attention to here is that humans are the purveyors of science. And so humans make observations, run experiments, and we try to learn how natural processes work. But on this tree of life, humans have only been here for a little while. And if humans are there to make the observations, and we've only been here for a little while, then everything beyond that, back into the past, we don't have very good observation of. I'm not saying that science can't touch this, but it's foggy. It's less clear. You have lower confidence when you come back here. And then I think it gets interesting because you can see that the part that humans have observed is actually extremely similar to what this hypothesis also shows. And so it's fundamentally difficult to tell these apart because what humans have observed is really quite similar. And so we're challenged then to try to tell them apart. But if you're a good scientist, what you do is keep an open mind, push your biases aside, collect some evidence, and let the evidence tell you, which hypothesis is more favored. And of course, you have to keep your mind open because over time, new evidence may come and maybe could change your mind. Who knows? But that's what we're going to try to do. And I'm sure you're all very familiar with the fact that most scientists, biology books, most media will very clearly tell you that the evidence overwhelmingly supports evolution and only a fool would disagree. And so I'm here to tell you that I have three big problems with what's going on here. The first big problem is that, things like the biology textbook and lots of media today, they will present a very much one-sided argument where all the evidence, every piece of evidence is in favor of the tree of life and macroevolution. And there's no limitations, there's no counter evidence, no concerns. It's just all proven. And that's concerning. Second thing that bothers me is that oftentimes evidence that is actually equivocal, evidence that could equally support both hypotheses, is presented as always being in favor of evolution. And to give you an example of that, Kirby mentioned some videos I've done with Jim Torr. We've had a long back and forth with some strong evolution supporters in a series of videos. But their main argument was that looking at the DNA across various life forms, they see strong correlation or a nested hierarchy, you could say, in the DNA evidence going across life, meaning that there's a lot of shared aspects of DNA across life. And they say that is exclusively in favor of common ancestry. And they even said it so strongly as they said that it disproves separate ancestry or the forest of life. But when you think about that openly, having shared genes in different types of life, that could be evidence for common ancestry, but it could also very equally be evidence of a designer using the same parts or reusing parts. I'm a designer, and every designer I know tends to take the previous design and add a few innovations, a few tweaks to it to come up with the new design. And so there's naturally a correlation structure to the process of design. So that evidence, which I think is equivocal, was being used in support of evolution. And I don't think that's correct. And the number three big problem here, and this one may be more subtle, this is we're going to spend more time, but they often take evidence that only gives low confidence and prioritize that over evidence that actually comes with high confidence. And so you might be wondering, what do I mean by these terms if you haven't heard them before? I work in the field of medicine, actually in heart failure and in cardiac disease. And medicine, of course, is a life and death for all of us. All of our lives have been impacted by medicine. And so because medicine is so important, we even demand that medicine has to be supported by the best of science. That doesn't mean that it's perfect or that it doesn't make mistakes, but it's so important to us that we really want the best of science. And because of that demand of society, medicine in response has established well-known levels of evidence where all the doctors have gotten together and said, this kind of evidence up here, we're going to really put our trust in, and evidence down on the bottom of the scale is kind of sketchy. So if a drug gets approved by the FDA or some procedure is approved for use on humans, you got to have really good evidence. And don't worry about the details here. This isn't really not about the details. It's just me saying that I'm very pleased that medicine has come to this agreement. They notice the difference between good quality and poor quality evidence. And you might find it interesting to read this down here, but the lowest level of evidence comes from a consensus of expert opinion based on clinical experience. So that's not given a lot of credence here. My wish is that all science, all forms of science, would have something like this, where you can prioritize evidence you could put more trust in, and then deprioritize evidence that's of lower confidence, especially evolution, I wish would do this. So I wrote this book to kind of bring this about, I try, but in this book, I talk about 6 criteria that are used to determine the level of evidence, level of confidence. And this applies to all of science. This is not just medicine. It could be astronomy. It could be transportation or energy or whatever it is, any kind of science. I believe these criteria help you to determine a level of confidence. And it's not a simple yes or no answer. It's actually kind of a spectrum of different levels of confidence. Very high, in the middle, medium, low, low confidence, okay? And I'm quickly going to go through these 6 criteria. I think you'll find them mostly very intuitive, but they help you to know how much you can trust some result. Like tomorrow, you're going to open the newspaper or see something on the news, and you'll hear some discovery. Maybe they found some molecules on Mars or something like that. And you can actually apply these 6 criteria to determine how much trust or how much confidence do I place in what I just heard on the news. So I think this is very practical. The number one criteria, everybody should be familiar with this, but if evidence is repeatable, you can put more confidence in it, right? So take a really simple example, like I'm going to drop an object and see how fast the object falls. I can do that, and then I could give you the object and you can measure it, and then someone in Japan can measure it, and we get the same answer. And every time you repeat it, your level of confidence gets stronger and stronger, right? That's pretty simple. That's number one. Number two is that you have more confidence the more directly you can measure a result. So to give an example of that, when I was a kid and I didn't feel well, and I would go to my mom, say, Mom, I don't feel well, what she would do is a very indirect measurement of my body temperature. And she would feel me out here and say if I would felt hot or cold or something. That's obviously not as accurate, not as confident as if you do a more direct measurement of temperature, right? This is more direct, and you get an actual number, which you can compare to 98.6, right? You have a higher level of confidence. So that's what I'm talking about with number two, directly measurable. Criteria number three is prospective study. So this is about planning your experiment in advance, writing it down and planning it especially so that you can block out confounding factors which can get in the way and confuse the result. You want to get everything out of the way so that you can actually determine what is the reason for what you've discovered or what changed here. So prospective study is much better confidence-wise than the opposite, which is retrospective study. Retrospective study is looking into the past and trying to figure out what happened, kind of like a murder case, a court case. It's much more difficult to be confident if you don't do it prospectively up front. So let's imagine that I am going to drop an object and see how fast it falls, right? Very simple science. I could look on YouTube and see lots of videos of things that drop, and try to measure that. But because I don't have control over the situation, because it's in the past, I mean, maybe there was a strong wind, or maybe they actually had the video upside down when they took the video, right? Where if I do it prospectively, I design the experiment. I can even like take the air out of the room, right? So it's a vacuum, and I have a much better unconfused measurement of how fast the object is falling. Prospective study is great. Number four, you want to avoid bias. I think you're all familiar with this one. We all have bias. I have bias. Everybody does. And it's kind of like a pair of, sunglasses, and you're looking through everything you perceive comes through your bias, which is the sunglasses. And when I do a clinical study at my work, I actually have to have a segment of the protocol I write on how I'm going to try to block out bias. And it's an active process. It's not just me claiming I'm going to try not to be biased. You actually have to try to take steps to make sure that bias can't creep in and ruin the result, because it very often does. Number 5 out of 6 is to avoid assumptions. Assumptions are basically shortcuts. And everybody tries to take shortcuts. You want to save money, you want to save time. Any kind of project, you have a budget and you have a timeline. So you want to take, you need some assumptions, but you have to be careful in what you choose as assumptions and be very open about those assumptions. Show people that you've taken those assumptions and justify them. So if I'm going to drop an object and see how fast it falls, a common assumption might be that the air in the room is just not a big influence. It's not going to matter that much. Because, taking the air out of this room is going to cost me 6 months delay, and it's going to cost $500,000. So can't we just assume that air in the room doesn't matter? So you can do that if you have some justification and you're open about it. The worst is when people hide their assumptions or aren't even aware of the assumptions they've made, because that will ruin good science very commonly. And then the last one, number 6, is about making reasonable claims. This one is more when you're done with the work, you've collected your data. Now, what are you going to say? How are you going to conclude something from your work? And you want your conclusions to apply to what you've actually studied and not stretch beyond that. I like calling this, you don't want to get out over your skis. You want to stay on your skis in concluding from what you actually did study. So I like this example here. You might find this kind of funny, but I like this example of a can of nuts. This can of nuts is something you could actually buy today, but they want you to buy more of these nuts. And the way they do that is they say they're heart healthy, and the American Heart Association has certified these nuts. Well, the FDA comes in and they say, well, what evidence do you have if you're going to claim that these nuts are good for you? And I really kind of like the statement they have here on the can, because it is a reasonable claim. It says, scientific evidence suggests, but does not prove, that eating nuts may reduce the risk of heart disease. So you can see there's some hedging language here, and I think it's a very balanced conclusion. I think it's a fair assessment of what their data actually had to say. And I say that because I contrast it with, this is my son's high school biology textbook that he had, a very popular biology textbook. But it said in there, for example, organisms as dissimilar as humans and bacteria share genes inherited from a very distant common ancestor. So hopefully you see a contrast here between these. This one has no hedging language. It could say, we think, or we suspect, or they may share genes. This one is over exuding confidence in something that it probably is not really all that confident of. And it concerns me when I see this, because it surprises me. It seems like our society is more concerned about claims that we make about nuts than claims that we make about evolution. That these textbooks can get away with strong statements like this with very little supporting evidence. So we're back to this, where we're going to put on a spectrum a given piece of evidence according to those 6 criteria. And we've already been through the example of, if I want to answer a question, how fast does an object fall? Very simple question you can ask of science. Science can just knock this out of the park if you do it well, because you can get your result repeatably, directly observe the result, study it prospectively. You can be careful to avoid bias. I don't think there's a lot of bias inherent in this question, because it's not like I'm going to make $1 million, or get a promotion or something if I get an answer in particular here. Careful to avoid assumptions, like taking air out of the room, and then making reasonable claims. Here, there's a chance to, I could fail this criteria if I measured in my room here how fast the object falls, and then I claim that on the top of Mount Everest, I would get the same result, right? That would be getting out over my skis. It would be making a conclusion that I didn't actually study myself. Because in fact, the gravitational force is a little bit different, a little bit less on the top of Mount Everest. Anyway, let's contrast that, because this is a very high confidence kind of result. Let's contrast that with this statement from my kid's biology textbook. I just read it. Organisms dissimilar as humans and bacteria share genes from a very distant common ancestor. This statement is related to a question: do humans and bacteria share genes from a common ancestor? So, how much confidence can we have about this? Well, can you repeat this? In other words, take the common ancestor of bacteria and humans and watch that common ancestor turn into bacteria and watch it turn into a human. Well, of course not. It can't be repeated, not even close. Can you directly observe that process happening? Not even close. Nobody witnessed the actual event. Of course, it would take an incredibly long time to do that. Can you prospectively study it? Can you put this single single-celled organism in the laboratory and watch it turn into a bacteria, watch it turn into a human. Of course not. Can't do that. And I think you and I know that there's a lot of bias behind what people think about this statement. Right up front, lots of bias. What about assumptions? Are there assumptions here? I actually think the entire concept, this entire statement is an assumption. The assumption behind this is that if two organisms share a gene, then it must have come from common ancestry. It must have been inherited from a common ancestor. So the whole thing is basically an assumption. And as far as overstated claims go, we already talked about that, where there's no hedging language here. This is exuding confidence for something that apparently it's clear it doesn't have a lot of confidence. So this statement belongs way over here on the low confidence scale. I hope this is resonating with people. So back to our question of which one of these is correct? Which hypothesis is correct? We got to get the data. We're going to talk through some data. And basically, we want to prioritize data that comes with high confidence and deprioritize data that, of course, has low confidence. So what do we have for data? What kind of evidence do we have in support of evolution? The tree of life, single tree of life. Well, if you look at any biology textbook and pretty much any argument about in favor of evolution, you're going to find these four particular types of evidence. I'm sure you're very familiar with the fossil record, geographic distribution. This is why certain living things are in certain places on the planet, vestigial organs, and then a comparison of life forms. This is sometimes called homology. It could be you're comparing DNA across life forms, or even comparing like the structure of your appendage across life forms. And I wanted to point out, just to make things interesting, I have a little video clip from Richard Dawkins, and he's going to talk us through this and tell us which of these he finds to be the most compelling case for common ancestry. So let me pull this up. I hope you can see this and hear this.
Out of all the evidence used to support the theory of evolution, what would you say is the strongest, most irrefutable single piece of evidence in support of the theory?
There's an enormous amount of evidence from all sorts of places, and it's hard to pick one strand which is more important than any other. There's fossils, there's the evidence from geographical distribution, there's the evidence from vestigial organs. I think, to me, perhaps the most compelling evidence is comparative evidence from modern animals, particularly biochemical comparative evidence, genetic, molecular evidence.
Okay, I hope that came across pretty clear. But you basically heard Richard Dawkins bring up each of these four categories of evidence, and he said that the most compelling of all was this comparison of life forms, comparison of DNA across life forms. Well, maybe you'll be interested in noticing, therefore, that the statement that we just dissected, organisms as dissimilar as humans and bacteria share genes from a very distant common ancestor. This statement, we just dissected it and said this is very, very low confidence because it fails all six of our criteria shown here. That statement is exactly what Richard Dawkins is talking about. In other words, the best evidence he's got, the best evidence he can come up with is very, very low confidence. It fails all of these criteria. That's kind of concerning. And in my book, I go through all of the evidence, but I'm telling you that each one of these, all of these are all very low confidence evidence. And it's all taught to our kids as if it's the most compelling evidence. So it leads you to ask then, I'm assuming you're asking, well, is there any high confidence evidence for evolution? What is there that is able to give us some confidence? And I'll tell you there is. So we're going to go through some. And all of these forms of evidence, this is kind of like the title slide I put up, these all come from long-term evolution experiments where people like Richard Lenski plan the experiment in advance. It's a prospective study. It's repeatable. They're directly looking at DNA changes that happen over time. And they fit these criteria for high confidence if they avoid bias, avoid assumptions, and make reasonable claims, right? It's a big if. You got to get those three things too. So after 33,000 generations of E. coli that Richard Lenski studied in his lab, and I got to give him a lot of credit because he's put over 30 years of his life into studying 80,000 generations of E. coli. It's an incredibly persistent study that he's done. But after 33,000 generations, maybe you've heard this before because it was a big news, it was a big deal. The E. coli developed the ability to metabolize citrate, which is a molecule that was in their daily bath, their daily media, but it was there as like a chelating agent. It wasn't intended to be a source of nutrition, but the bacteria started to metabolize it and they grew faster and stronger because, they had another source of food. This is a big review article in Nature that just came out this year, but they concluded that the evolution of citrate utilization was a major innovation in the context of the long-term evolution experiment. And our friend Richard Dawkins, he said it's a beautiful demonstration of evolution in action. It's new information entering genomes without the intervention of a designer. So let's look into it because it sounds pretty impressive that evolution could accomplish this. Well, some of you may know, but it turns out that E. coli already had the ability to metabolize citrate. In fact, almost every living thing has the citric acid cycle, which is the ability to metabolize citrate. The challenge is bringing the citrate into the cell. And E. coli already had the ability to bring the citrate into the cell, but only if there was no oxygen around. So what evolved here, you could say, I guess, is that the ability to bring citrate in when their oxygen is present. So Lenski's group later discovered that this key innovation, the ability to metabolize citrate in the presence of oxygen, it actually required only 2 duplications of existing genes. One gene is an antiporter on the cell membrane that trades off bringing in a citrate for kicking out a succinate. So it's able to bring some citrate in that way. And it duplicated this gene and placed it after a promoter that is active in the presence of oxygen. So now when oxygen is present, it can bring citrate in. And when oxygen is not present, it can bring citrate in. And the second duplicating gene was this DCTA. And this is a membrane-based importer of succinate. So the cell could bring in the succinate and then trade it off for a citrate. And that's how it's able to bring in lots of citrate, even in the presence of oxygen. So that's it. I mean, that may sound complicated, but it's only copying 2 existing genes. So it's not new information, and it's not what I would call a major innovation. In fact, it's even a little worse than that. Gene duplications, like we're talking about, are basically the most frequent kind of mutation that happens in life. It's about 1,000 times more likely than a point mutation. Often when people talk about mutations, they're talking about a single nucleotide changing, from a guanine to a cytosine or something. About 1,000 times more likely that you'll end up just duplicating an entire gene. And this is a quote from a paper talking about how frequent duplications are. And the need for only these 2 mutations to give E. coli the ability to metabolize citrate was also shown later by Van Hofwegen et al. And they had a really cool conclusion in their paper. Here's the reference. They said, a more accurate, albeit controversial, interpretation of the long-term evolution experiment is that E. coli's capacity to evolve is more limited than currently assumed. It's kind of a controversial statement there. Not only this, but the E. coli that can now eat citrate in the presence of oxygen, they're not actually evolving toward a better future. They're actually devolving. This is a recent paper here by Blunt from 2020. And what's happening, actually, as I mentioned, is that formerly the bacteria had the ability to control when it produced this citrate importer, but now it is what they call constitutively expressed. In other words, the E. coli is producing this citrate importer no matter what, whether there's oxygen or whether there's no oxygen. And constitutive expression is not always very efficient. It's not always good for an organism. You want to have control over, the operation in the cell. So this paper they found, however, we serendipitously discovered evidence of substantial cell death in cultures of the E. coli that can process citrate, basically saying these results indicate that the evolution of aerobic growth on citrate in the long-term evolution experiment was associated with elevated mortality. So it's not actually much of an innovation, and it's actually maybe more bad than good. So when we come back to our big question, we got to keep coming back to this 30,000-foot question we're trying to answer. Which of these hypotheses is more supported? And again, we're going to pull up data, look at the data. So many people have claimed that the long-term evolution experiment of Richard Lenski is strong support for common ancestry. But with all that we've just talked about, when we actually pull up the facts and dig into the papers, I think I can make a pretty good argument that this is actually more supportive of separate ancestry here, because you have 80,000 generations of E. coli and you end up with E. coli and a few tweaks, like a few duplications of existing genes. If this hypothesis was correct, you would expect, because this single cell that started life and moving out to be all of modern life, there's millions of innovations, dramatic innovations that have happened to make modern life. So you would expect 80,000 generations of studying a life form, you'd expect to see something innovative, something cool, something new, some new information. And we're just not seeing that. So it doesn't support this tree of life. So we have a few more studies to go through. This is actually my favorite one here. And here's the reference. But these investigators, they actually purposely went after a test to see how much evolution could or could not accomplish. It's pretty cool. So E. coli, we're back to our favorite little bacteria. E. coli have the ability to produce their own tryptophan, which is an amino acid that all life needs. And they use this enzyme tryptophan synthase to do that. So these investigators said, hey, if we intentionally damage the gene to make tryptophan synthase, actually two genes, if we intentionally make a change in a nucleotide to damage it, can evolution repair that damage? Kind of a cool study idea. So they changed one nucleotide, one single letter out of about 2,000 letters of DNA to make this enzyme. They changed that one letter and they put this mutated E. coli into a bath that had a limited amount of tryptophan. So they're applying some artificial pressure that if you can't produce your own tryptophan, you're going to be suffering. If you can produce your own tryptophan, you can win, you'll take over. And what did they find? Well, after only about 100 million E. coli were grown, which is not much for a bacteria, the mutation was actually repaired. So they believe that a random mutation flipped the nucleotide back to the one you want, and now you have fully functional tryptophan synthase, and it took over the population. So that's success. That shows you that evolution, random mutations plus natural selection can fix this kind of damage. And they said, okay, let's do it again. So they picked a different single letter of the DNA and made an error on purpose there and put the mutated bacteria in the solution with limited tryptophan. Now let's see if it could fix this one. And they got the same result. After about 100 million E. coli were grown, the bacteria evolved to fix this problem and then took over the population, had a happy ending. So that's not all they did. They did one more experiment, and maybe you're thinking about this already, but they said, what if we do both of those changes and see if evolution can fix that problem? And in this case, the enzyme became completely not functional. With the single error, it was somewhat functional. And now with both errors, it was not functional. And so they took that mutated E. coli, put it in a solution with a limited amount of tryptophan, and they actually went through 9,300 generations of E. coli, trying to find an E. coli that would fix these 2 point mutations. And that's about 1 trillion organisms that they grew. This is far more than all humankind has ever had as far as numbers. And what happened was the function of tryptophan synthase was never restored. In fact, in some of the genomes, the gene was jettisoned. It was just gone. In some other genomes, it was just a suppressed production of the gene. Why is that? Well, in order to produce this enzyme, you need a lot of tryptophan. So if the bacteria were trying to produce a broken enzyme, they were wasting some of the limited tryptophan they had. And it was actually detrimental to try to produce this if it wasn't working. And so some of them actually jettisoned the gene. I find that very interesting. It kind of poses a limit on what evolution can accomplish. And to put that in particular terms, evolution in this case failed to repair an enzyme that was already 99.9% fully intact. Specifically, it's about almost 2,000 nucleotides of DNA in these genes, and there were just 2 nucleotides that were wrong. So that's why it's 99.9% intact. And evolution couldn't finish the job. And so how is it possible then that evolution came up with this gene from scratch, or even from a similar-ish gene, if it can't finish the job given 99.9% complete DNA? Very interesting. So we get back to our 30,000-foot question here of which of these is better supported by high confidence evidence. Now, I guess I haven't been emphasizing this enough, but if you think through that study I just talked about with tryptophan synthase, it was repeatable. They froze some of the bacteria, they froze them during the experiment that they could repeat the study, and they had multiple aliquots of DNA of E. coli during the process. There were multiple ones. So it's easy to repeat. It was prospectively studied. They directly measured the DNA changes over time. And then there's bias, assumptions, and making reasonable conclusions. Anyway, so in this case, I think it's pretty clear that this tryptophan synthase experiment, just like the long-term evolution experiment, is favoring a forest of life, because we're not seeing the kind of ability to innovate and to produce, very diverse modern life today from a single-celled organism through common ancestry. We just have a few more cases they're going to go kind of quickly. But like the long-term evolution experiment, this group, here's the reference for it, they're interested in studying multicellularity. In other words, how did evolution go from single-celled life to multicellular life? Certainly a critical transformation that happened, and they'd like to study that. So they did this study in yeast, which is eukaryotic, and yeast reproduces by budding. So you get this kind of a little blurb here that, starts to build up some of the cell contents and get a duplicate of all the contents of the cell, and then it pinches off, and you have a mother and daughter. To start this experiment, they actually deleted one gene in the yeast, and it's this ACE2 transcription factor. They took that gene away, and it was already known before they started this experiment that that deletion causes what's known as snowflake yeast. And this is yeast that's already multicellular, and it's because the mother and daughter are no longer able to separate. It's kind of like the umbilical cord can't be cut anymore. That it pinches off, but they're not able to become two separate cells. But they took this snowflake yeast and they went through 3,000 generations where the bigger clusters would sink to the bottom of the test tube, and they would take the biggest ones and use those in the following day's experiment. So it's artificial selection to see how big they could get these clusters to become. And over 3,000 generations, they found that they had evolved, quote unquote evolved, to become tens of thousands of times larger cells, bigger clusters, and they became tough, like tough as wood. So here's the ancestor of the snowflake yeast little thing, and they got them to become quite large. And I used, quotes around evolved here because in the papers they didn't yet talk about what had changed. What kind of DNA changes has happened? Is this really an innovation? And it's quite interesting because in this paper, what they end up showing is that by deleting 3 genes, this is, they did a little aside on the experiment and they genetically engineered these yeast and took away 3 genes. 2 of the deletions made the cells like longer and thinner, and 1 of the gene deletions increased the strength between the mother and daughter bond. And they said they were able to engineer the development of similar macroscopic clusters. Here's a quote from the paper. Engineered strains with mutations increasing cell length and bud scar size recapitulated our evolutionary progression. So in other words, this graph shows how big the clusters were. Their goal was to get really big clusters, and also the aspect ratio of the cell. And the little gray dots you see here is the artificial selection through 3,000 generations. So they're getting bigger clusters with longer aspect ratio after 3,000 generations. But then they said, oh, we just deleted 3 genes in addition to the one we started with deleted, and they end up over here, which is pretty much the same result. So we're not seeing, it's not totally clear what genetic changes happened, but it is clear that simply deleting some genes gets you almost to the same accomplishment of the experiment. We're not seeing a lot of innovation here. Once again, if this is true, we'd expect to see over 3,000 generations, we'd expect to see something innovative going on. But in fact, what we're seeing is yeast stays as yeast. And in fact, the multicellularity is something that they forced upon the yeast because it couldn't divide, and it would probably prefer to divide, but it couldn't. Okay, one more case, one more study. These folks put something like 18 years of their lives into studying fruit flies, Drosophila. And they went through 600 generations of Drosophila. And once again, it's this high confidence evidence, prospective study, replicate populations, and direct measurements of the DNA changes. And in this case, they selected each generation, they selected the Drosophila that hatched earliest and matured fastest. They picked those to mate for the next generation. Everybody else got thrown away. And what did they find? Despite decades of sustained selection in a relatively small sexually reproducing laboratory population, selection did not lead to the fixation of newly arising, unconditionally advantageous alleles. In other words, that's a fancy way of saying nothing really changed in the DNA. Which is a shame. You've put in 18 years of work into something, and that's kind of the conclusion you get. And then they go on to say, this is notable because out in the wild, we expect the strength of natural selection to be less intense than this laboratory artificial selection. And the environment is unlikely to remain constant for 600 generations. So consequently, the probability of fixation or lots of genetic change happening in wild populations should even be lower than the likelihood in these experiments. Very interesting. So once again, our big question here of which of these is better supported by evidence. I think the Drosophila study is also telling us that living things tend to stay within their tree, and there's not a lot of innovation going on here. Okay, so bringing it together now, we've gone through these 4 experiments, and I'm telling you, I have another 10 experiments like this that give basically the same picture. We just don't have enough time to go through it, but they're all giving you the same kind of picture that evolution is a very weak, very constrained process. I would say it seems to be incapable of producing the kind of innovation that we all know in modern life. So what we're doing in these experiments, these high confidence kind of experiments, is we take a living organism, whether it's a Drosophila or an E. coli or something, and over a long time period, we subject it to selective pressure, whether it's artificial or natural selective pressure. And basically, it has to either change or die. So it either changes or it's going to be thrown out or die. And you do that generation after generation after generation. You're trying to force changes to happen. And then you look at the DNA that comes out of, compare, you can easily compare the final result to the original result and look exactly what happened here, what changed. And so you've seen in the studies from Richard Lenski, from the tryptophan synthase, that very minimal changes have happened. So now we're going to apply this. This is where it gets really interesting. If you take what we've learned here and apply it to this scenario, okay? We've all been taught that the common ancestor of chimpanzees evolved into humans and chimpanzees. Is this something that could be accomplished in something like 6 million years, given what we've already known about what we already know from high confidence evidence about what evolution can accomplish? Well, it's very interesting because a recent paper talks about this. And this recent paper by Yu et al. Came out in 2025. You've probably seen a lot of reports about this. But this is the most complete sequencing of genomes of chimps and humans that we've ever had. And they go into quite a lot of detail. But if you look at these figures in the paper, it very clearly shows that comparing the two genomes, that 13.3% of the human genome, if you try to take that DNA and look through the chimp DNA and find a matching segment, there is no analog. There's nothing that matches to it. And they call this gap divergence. And if you take pieces of DNA where there is a nice sequence map or analog, the difference between the human and chimp DNA is about 1.6%. This is probably a number you're pretty familiar with. This has been tossed around for a long time, but nobody previously really talks much about this, 13.3%. But when you do the simple math here, because our genome is like 3.2 billion nucleotides, this 13.3 plus 1.6, it gives you something on the order of 470 million nucleotides of DNA that separate us from chimps. Quite a large amount of information, quite a large amount of DNA. And so you put that into perspective with what we have learned from these long-term experiments, from this high confidence evidence, what we've learned about the changes that can occur, and then say, do you think in 6 million years, given pretty low reproduction rates, 15 to 20 years reproduction over 6 million years, do you think you could accomplish 470 million nucleotides of DNA change here? Doesn't seem possible to me. It seems very, very far from possibility when you focus on the high confidence evidence. So in my view, and what I see in the data, comparing these two hypotheses, we end up with this kind of scenario. A lot of the low confidence evidence is equivocal. It could be supportive of either one. But the high confidence evidence, I think, stands very clearly in favor of more of a forest of life. So that's where I'm going to end. I'm going to end by showing you a couple of books I've written. This one is more about the origin of life. And I have a co-author, Change Tan, who's a professor of biology. And I think you'll find that interesting. And if you don't like reading, you can go online, go on YouTube and look up Long Story Short, Origin of Life. And there's a whole series of animated, fun-filled videos that go through why life did not just start spontaneously through natural processes. And I'm open to answer any questions. Here's my webpage and my email address if you have any other questions. But thanks for your time.
Barry, I have a comment.
Okay, go ahead, Lothar.
Well, first of all, I think this was one of the most compelling um, seminars that I've heard. It's very similar, I think, to one by Cy Gart. Are you familiar
Yeah.
with him? Yeah, I
Yes.
think he thinks the same way. Now, on the fossil records thing, I don't think anybody's seen transitions. So I don't think those fossil records explain anything. And
I
then there's finally, many years ago, I read a book by John Sanford on genetic entropy,
hmm
and that kind of clearly tells you that Darwinian evolution just can't be true.
Yeah, I agree. Thanks for bringing those up. On the topic of Cy Gart, um, if you're a fan of Jim Tour,
I
if you go
am.
on, yeah, if you go on Jim Tour's YouTube channel, just last week I think he released an interview with Cy Gart.
I did see it.
Yeah, and it's fascinating in there because at one point Cy Gart admits that he had believed that gene duplication was responsible for lots of this innovation, like even making an invertebrate into a vertebrate. He thought that happened through genome duplication. And Doug Axe called him on that. Doug Axe said, "Why don't you look that up?" And you can see an interview. It's like at 54 minutes into this interview that Sygart said, "You know what? I did. I went and I read 10 papers and I couldn't find any evidence that that genome duplication led to innovative
Mm.
DNA changes." And he said, my jaw dropped, and Doug was right. And I give him so much credit for saying this online and being open and honest and, you know, winsome. It's very cool.
Yeah, very nice. Thank you.
Mhm.
Anyone else?
I had a question here. Mine might be a little complicated, and I don't understand biology well enough, but I was having a discussion with a friend who's convinced of the whole macroevolution scenario. And here was kind of his argument for why one of the evidence he sees. He says humans have over 100,000 specific inactive virus fragments. He calls them ERVs, whatever those are, in our DNA in the exact same location where chimps and bonobos have them. And he says these represent rare times when a virus inserted itself into a sperm or egg's genome, but the cell survived and the allele became permanent in the population.
Do
So,
you see any assumptions there?
well, I was
That's
going
the
to
key
ask
question.
you to kind of tear this apart for us, and so
Well,
he's assuming the cause. I don't know that we know the cause is viruses
right.
inserting.
That's right. Right. So fundamentally, what he's arguing is based upon assumption.
Yeah,
And you could push back if you like to say, how do you know that that assumption is valid?
all right. Yeah. Now, if these things are non-functional, which is what he implies,
That's an assumption. Yes.
yeah, I guess that's true too. Um, Inactive virus fragments. So,
Mhm.
uh,
Junk DNA.
well, I don't think this is quite junk DNA, but it might be related. Um, and again, I don't know enough biology to know whether it is or not. But Kirby, are you familiar with these arguments? Or
Yeah. hmm
okay,
I was wondering if the 13.6% of the uh, difference between the chimp and the human genome was mostly these ERVs. I'm assuming it was actually, but do you know, Rob?
No, I don't know the statistic there, but I think this person is arguing that because chimps and humans have the same
ERV.
ERVs in the same location, that those are not differentiating. That's
Right.
not part of this 13% difference.
Yeah, no, this wouldn't be a difference then. It would be a similarity.
Similarity, yeah. Yeah, I think I've heard arguments like that, and I think they need to put their assumptions down on paper,
Uh-huh.
and start by focusing on those, because otherwise the argument they're trying to make is based on a house of cards, if the
Yep.
assumptions aren't real.
Yep. This is Jay.
Randy Lewening, you're next.
Okay, good. No, thank you so much. This is a great discussion. Two things that sort of jumped out. One is sort of the picture of a product designer, and so if you look at General Motors, you'll see a whole bunch of different types of cars, and they do share components, but it's not because one car evolved from the other. It's because it had a designer that had a common technology and design philosophy, or Boeing or anything else. So I guess one question is just understanding, what we see in what we observe in nature in sort of that context. And the other thing is something that we often see in nature, which someone could which has some similarity to evolution, but I think is different, is where you've got a population that has natural variation, and and one sort of subspecies survives and thrives because of a change of environment. And an example, like in Martha's Vineyard, where I just was on vacation. Basically, you have you have some variations of clams and some of them are more resistant to a particular disease than others. So you can, as a marine biologist, either try to repopulate the more resistant version, or the more resistant version may ultimately survive and flourish. But that's not because anything's evolved; it's because the variety was already there. So anyway, maybe you could say
Yeah.
something about each of those observations and how they fit within this framework.
Well, I think I really like what you're saying there, and I think this goes under what I kind of mentioned, is that observations like what you just brought up are often given credit, or they give credit to evolution for that, when either they're equivocal on both sides, both different hypotheses, or maybe even they're more supportive of the designer hypothesis, because you actually have the designer building in mitigations for environmental changes. Like living things have been given a toolbox that they can use as resource when the environment changes and they face some challenge. They pull out the tool that'll help them to survive. I think that speaks to a designer. But the evolution camp will say, oh, they evolved that ability because evolution is infinitely plastic. It can explain anything while explaining nothing. So they'll take your example and say that's a sign of evolution, but I think it's much more a sign of a designer. And the more sophisticated the mechanism is, the more clearly it is, I think, evidence of a designer. When you see, like, for example, maybe you've heard of this, but there's a big, long 10-year study about rice in warm climates. When they take rice and move it to a cold climate, it has trouble producing seeds. In other words, food, right? So it's very important that rice produces food in cold climates. So they studied it for 10 years, and they found that after a few years, they could get the rice to produce food in the cold climate. And they traced it all the way down to the cause and effect of what caused it. And it's a brilliant paper to read. But basically, what it is epigenetic changes. It was methylation of one gene that, if the methyl tags are there, the gene is not needed, it's turned off. And in warm climates, the methylation tags were there. And in cold climates, the methylation tags had to be removed without changing the genome at all. And the rice was then able to survive in cold climates. So it's almost like, when your mother used to send you out on a cold day and you'd have your jacket on, but you started shivering. You reach in your pocket, oh, there's a pair of gloves in here, and there's a hat in here, because your mother prepared you for this stuff. And it's kind of like God, you could see God preparing the rice for this environmental challenge. Here's your mittens and here's your hat, and you're going to be fine.
I need to inject a question here. I haven't heard anything from Dawkins or anyone else that has an explanation for epigenetics, which was what you were just talking about.
Yeah, well, it all evolved. That's the
How?
explanation.
How? Okay, so
Yes.
let's go on to Tim Boyle. I think you're next. You're muted, Tim. You're muted.
There
Can
it
you hear
is.
me okay now? Yeah. Yeah. The question I had to do with it's part of the fossil record issue, but I've heard, like Hugh and Fuzz mention a lot about mass extinctions followed quickly by mass uh, speciation. And it seems like I just kind of wondered in your reading and keeping up with that subject, is there any indication of really how fast that occurred? Any new data or anything like that? I mean, if it could be, just over a few centuries, or maybe it's a million
Hmm.
years or something. Any data on that?
Well, I'm not a paleontologist, um, and I can tell you, um, from the presentation I just gave, I think you could tell that I'm not a big fan of using a lot of fossil evidence because it
Mm,
doesn't meet these six criteria.
mm,
And when evidence comes with low confidence, like a lot of fossil evidence, you can kind of make a story up and people can't really defeat your story. So you end up arguing forever and ever and ever and never, coming to any agreement.
mm,
I'm not saying that all of paleontology is low confidence. It depends on how it's used. For example, if I wanted to say um, the size of a T-Rex's femur is 3 meters long, that's something I can repeatably address, and I can see it again and again and directly measure it. So I can say that with confidence. A T-Rex femur was 3 meters long, whatever. But to apply fossil evidence to support evolution or a lot of conclusions that are made about fossils, they get pretty sketchy pretty quickly, I think.
mm. Yeah. Okay, that's good.
Ari Goldberg.
Um, Hi, Rob. First of all, thank you for tackling this topic of evolution. I think it's the most important topic in apologetics because of my own experience, partly, and I may get to that if I have time. But I'd like to ask you, so let's say an atheist were to come to you and say, okay, so you think evolution, life forms didn't happen naturally. You think God did it. So how did he do it? Would you be able to give them a rational explanation? Allow me to expand just a bit on what I mean. God sometimes uses natural processes in conjunction with miracles, like there was a strong east wind that helped part the Red Sea at the Exodus. There may have been a cosmic airburst that destroyed Sodom and Gomorrah. There were earthquakes and eclipses that coincided with the crucifixion of Jesus. Um, so, if God were to write a report on how he created life, or if you had been there while he did it and were able to ask him any questions, what would he say? What would the report say? And I would like to add that something Jim Tour said puzzled me when somebody asked him basically that same question. And I believe he said that if we give it enough time, if it could take centuries, but I think science will eventually figure
Mhm.
out how
Mhm.
how life originated.
Yeah, how life began.
Mhm.
So
That is a place where Jim and I deviate a bit. Every time he says that, I cringe a little bit because I see it a little differently. But I think Jim's trying to say that we can't shut down science. and you can't just give up, that part I agree with. But I tend to think that we've already learned enough about how natural processes work, and we've already learned enough about what life requires to know enough to say that natural processes don't naturally produce life. That's an origin of life question. Um, but I think Jim and I might disagree a little bit on that. Um, but your first part, does that help, or you had a different
Yeah, in
first?
other words, what it seems like it requires some kind of miracle, like turning water into wine or multiplying the
Mhm.
loaves and the fishes.
Yeah,
So, in other words, if you could watch God create an organism from scratch,
yeah,
would you be witnessing God just um, drawing molecules out of the primordial mud and
yeah,
and string them together,
yeah,
or would he just snap his fingers and it already be there? Or what do you think?
yeah. I wish I knew.
How would you answer an atheist if he kind of pressed you in terms of like, well, how do you think God did it? Do you believe in
Right,
magic?
right. And you hear that often. Um, it's a good question because you do hear it often. But I think the fundamental problem is that the atheist is thinking only in terms of materialism, naturalism, right? And so they are constraining their thinking, and they're unable to realize in that case that God is beyond matter and beyond space and beyond time, where we're constrained by being part of matter, space, and time. And so if they are demanding that this creative process that God did, if they're demanding that that fits within their constrained world where we have matter, space, and time, then that's practicing a kind of theology, right? It's a kind of theology. I would call it a kind of a straw man theology that is constraining, and it's not my God. Maybe that's their God, but that's not my God. Um, and so they're not going to come to an answer if it's constrained to be a natural process. That's where they're at.
Right. Okay. Thank
You
you.
have to humble yourself and say that, okay, my science is limited, I can use my science to do all kinds of cool stuff, but I will not be able to explain how those molecules came together. It's beyond me.
Right.
Ken Wolgamuth, your turn.
Okay. Hello, Rob. I have a question about chance and then a question about the fossil record. After we had that communication, I connected with Chance Tang a little bit up. Is she still up at University of Missouri, Columbia? And she was kind of nervous about how her Christian convictions might play out being there in a secular university. So that's my
Hmm.
first question. Second question in terms of
Well,
the,
maybe I could just answer that, but
okay,
she's
that's
now
fine.
she's now at Missouri Baptist University
Oh, okay.
because she was let go from University of Missouri,
Columbia. Okay.
even
Yeah.
though she was tenured.
Yeah. She was nervous about that uh, very clearly, and I never got a chance to try to find her again. Is that
Yeah,
still at Columbia, or is that over in St. Louis, or where's that university?
it's close, but I don't know if it's the same city.
Okay, that's fine. Then in the fossil record, but in your book, you said, as I recall, I haven't read it for about 10 years. Um, you affirmed that the fossil record has credibility because we can go out and look at it over and over and over again. And in doing that, at the end of the Permian, the families of species dropped from about 400 to something like 200 families. And then from your perspective of the biosphere, what applies to the rejuvenation of having so many new species that have come about after that time through the Mesozoic and the Cenozoic?
Well, I think I mentioned already that I'm not a paleontologist, and because of, these criteria that I focus on for focusing on high confidence science, and generally the fossil record is more low confidence, I have trouble making a lot of strong conclusions out of the fossil record. But I would not say that speciation is, I mean, speciation can be observed. I don't think it happens commonly, and I don't think it necessarily involves a whole lot of innovation. But the development of speciation, I don't think is anything against what I've been saying. So if you say the species number increased, maybe that's so. But today, looking at modern life, speciation is defined as, a reproducing organism that it reproduces something, but now it's isolated from reproducing with other organisms, right? That's a separate species.
Yeah.
That's how we define species. When you go to the fossil record, how are you going to tell me that this thing could no longer reproduce with that thing? When you go to the fossil record, species are defined more by just simple morphology. You know that my tooth was thicker and bigger than your tooth, and therefore we don't reproduce any. It's really quite subjective. Sorry if there's any paleontologist online, but
No,
I
no,
find it all to be very
I'm
subjective.
not asking. I'm thinking about just the biological process. So you think potentially just ordinary adaptation could in fact be an adequate answer for that.
I think it can cause some speciation, but not like dramatic innovations. They just may be separate organisms that don't reproduce anymore.
Well, yeah, in the fossil record, we don't have DNA, so we don't have that kind of level of data. We just have the body forms, of course.
Right. And
Right.
they make a lot of judgments based on that. Yeah.
Yeah. Thank you very much for joining us tonight, Rob. Appreciate it.
Thank you. Appreciate your question.
John Yu, you're next. And you're muted. Now you're not.
Hi, Rob. Thank you very much for your very, very good talk.
Thank you.
Very good argument. I have just two parts. One is this origin of life research that's been going on for ages. I wonder, is this still going on? And if it is the excitement still there, or are the scientists, evolving into something that's just kind of boring?
Well, unfortunately, there is a lot of origin of life research going on, and in the media, you'll hear a lot of it. Every month there's some big story about, oh, we now understand how life began, or we now see this. There's always big claims, a lot of claims being made. And Jim Torr and I really try to focus on knocking down those big claims, because
Okay.
I think our society is being brainwashed by all this media hype that progress is being made. What's actually happening is the more we learn about what it takes to make life, I think the goalposts are moving further and further away every year between what natural processes are capable of and what's required to get life started. It's being separated more and more every year.
Okay. Well, that's interesting. So to hear that there's more activities in this origin of life research, more
Yes.
news being put out.
Yes.
So in your analysis tonight, you picked a few examples. I wonder if you could do the same thing with these new news that's coming out and measure the confidence level of those claims.
Yes. That's a good point. Yeah, I like that. I can point you to for years, this is getting a little detailed, but for years people believed that the foremost reaction explained where sugars came from, like ribose. You can't make DNA unless you have ribose as a sugar. And where do you get ribose at on the prebiotic world? Well, they said the foremost reaction did that. That was always the answer. And just about a month ago, a paper came out where they rescinded all that and they said, you know what? We actually do not know where sugars could come from on a prebiotic world.
Okay.
And I would consider that to be a high confidence statement because they did a lot of work to try to show
Well,
it, and they couldn't.
yeah. So it would be very interesting just to keep a record, ongoing record is sort of like between you and Jim Tour, and maybe some other people in the apologetics group, where you say, okay, here's a claim, and is it a high confidence? And you use the same kind of analysis. And so over the course of a year, maybe you will be able to have a running log of these things so that it would be kind of interesting.
Yeah.
But anyway, so my second part of the question or the comment is that is it possible to use the same kind of reasoning and methodology to argue the confidence level going the other way? In other words, start with a multiple tree of life, okay, on the right-hand side, right? And since you can't do scientific experiments like you could do in natural science, but there are other areas like historical records, fossil records, testimonials, those kind of things, and then evaluate that using a similar kind of yardstick and saying, okay, these are high level confidence. Is it possible? I'm not sure it is, but it would be kind of like a fair game because you argue from the left to the right balance. Now we want to argue from the right to the left balance to give the total objectivity.
So you're saying try to support the forest of life?
Yeah, the forest life, yes.
Yeah. Try to support that through experiment?
Not experiments, because since, you can't do, I don't know if you can do experiments, but there are other things you've cited, right? Fossil records, you've cited, then there is geological records, there could be historical records, those kind of things. It may not be the same kind of criteria that the tree of life would use, but you come up with this. It's very much like who's gentleman, very popular on the 13 evidences that most atheists would sign up to about the resurrection of Jesus Christ.
Oh, right.
Something similar to that, but not entirely, but more of a semi-scientific, objective kind of approach, like what you did, going from the tree of life to on a balance, but going the other way using the forest light.
Yeah. Well, thank you.
I don't know if it's possible, but it might be interesting if you can figure out one.
Thanks for the suggestion. I'm going to take a note on that and think about that some.
Okay, thank you very much. But I really enjoy your talk.
Thank you.
Ari Goldberg, you're next.
So about your books, how would you describe the distinctives or how they might differ? What kind of gaps did you try to fill with different books in that field, like by Douglas Axe or Michael Behe about the problems of evolution?
You're asking how the scientific approach to evolution book is distinct, you mean?
Well, yeah. What kind of niche or what kind of question does your book try to answer that makes it kind of unique from similar books in the field?
Yeah, I think, well, the scientific approach to evolution, it came from my career because I do medical research. And when I go to FDA and say, hey, we want this new product to be released for use by patients, right, by doctors, the FDA demands the evidence I provide to be of certain caliber. And I got used to dealing with FDA like that. And then I read my kids' high school biology textbook. And I was like, the evidence that's in there meets none of the criteria that I myself am held to if I'm going to sell a medical product. And it really kind of bothered me. And I started writing down, what am I held to, and what is going on in this biology textbook. And that's where these 6 criteria came from. And so this focus upon high confidence or low confidence, I haven't, I think that's to answer your question. I don't think I've seen that in other books. I'm sure it's out there somewhere because almost nothing is ever new under the sun. But um, I have heard people talk about distinguishing historical evidence from observable evidence. So that's kind of it's related, but I think this adds a little more formality to it.
Right. Okay. Thank you.
Yeah.
Can I ask another one? More of a theological question, I suppose. But in Romans 1:19-20,
Yeah.
Paul says that the evidence for God is obvious, so people are without excuse. And for myself, growing up in a totally atheistic environment, when I saw the museum exhibits in San Francisco showing, apes to man, kind of like the picture you showed tonight, I just believed it. And I never really asked myself any fundamental questions about whether there was how did everything get here, or what's the purpose of my life, or where do I go when I die. Um, and when I did read the Jewish prayer book, which had addressed God and talked about God, but I just automatically assumed, there's that word assumption again, that this was a holdover from a pre-scientific age, and so it was just pure mythology, and I could just ignore it, that there was anything to it at all. So it's basically where I'm going with this is it seems like, and it's not currently a struggle for me. I'm a strong believer, but it seems like evolution, especially when Dawkins said, atheism made it possible to be an intellectually fulfilled atheist. It's like evolution seems like it's a stronger deception, or a stronger um, idea, more powerful idea to bolster atheism than any preceding generation in history
Yes.
that I can think of. And so
Absolutely.
that's why I think it's so important what you're
Yep.
doing. And I'm just
Absolutely.
wondering if you agree with that, or how do you
Yeah.
how do you talk to an atheist that says, the Bible says it's obvious you're foolish to not believe in God, but
Mm.
I don't see that,
Right.
you know.
Yeah. That's a great question. I wish there was an easy answer for that. What I like to say, I think, is um, Charles Darwin's greatest accomplishment, I think, was that before Darwin, before evolution, everybody knew that the agency to create life and to make life belonged to God, right? God was the creator. And Charles Darwin knew that you needed some agency. You needed something to get the job done, the creative force, the agency. Charles Darwin stripped the agency away from God and gave it to this thing called natural selection. Natural selection is now the agent, and that's what does the selecting. Natural selection chooses, natural selection, prefers, or whatever it is. But that's kind of like a mystical, made up thing, because I don't think nature really picks like that. And the example I like to use is, maybe you saw back when there was a big hurricane that hit Florida. There's a picture of a whole neighborhood completely wiped out, just decimated. All the houses are gone, except right in the middle, there's this house that's perfectly standing. It looks as good as new. And so that house was designed to be hurricane proof, and it proved that it was hurricane proof. It survived. But no one would look at that picture and say, nature selected that house to survive the hurricane. It makes no sense at all. It's the designer who designed the house that could survive a hurricane. You know, the agency is in the designer. The agency is not in nature. Nature just caused a storm, you know. So Charles Darwin's big success, I think, was to strip the agency away from God. And we have to give it back.
Right, right.
May I ask another question?
Sure.
Oh, well, let's see. It's actually Kirk's turn, and then so Kirk, and then Ken, and then Lothar. Is that
Okay.
Lothar? Were you Kirk? Go ahead.
Um, yeah, so I was thinking about your what was it, 6 criteria, and you were saying that claims about evolution fail all 6, and I guess I want to push back a little bit on that. The repeatability, yeah, we can't go back and repeat the historical process, whatever happened, but we can repeat our observations and we can go look at new things that haven't been looked at yet.
Mm-hmm.
And the same thing on the perspective. You know, we can't, I think we can do a prospective study. We can say, well, if my understanding is correct, then I should find this in the genome. And I think in that way, it's similar to geology, which is also a historical study. You can't go back and repeat the geological process, but you can make theories and then go try to see if what you observe matches the theory or not.
Mm-hmm.
And I think the same thing would be true of astronomy, astrophysics, that we can't repeat a supernova explosion, but we can study what we see and then we can watch for a new one.
Mm-hmm.
And so I guess I'm curious what you think about this. And, you know, I think it was a little, I think there are some ways that we can do some repeatability and some prospective studies on claim evolution.
Yeah, I don't mean to make it completely sort of black and white. Um, what you have to do, I think, is write down the question that you're asking of science, and then you can determine if these things fit. Like, in other words, if I were to say, did a T-Rex exist? That's the question I'm trying to answer. And I can say, oh, I just dug up a T-Rex fossil, so that proves it. And I just dug up another T-Rex fossil. So I repeatedly tell you that a T-Rex existed.
Yeah.
And I think everybody would agree that's repeatable and yes. So I answered that question.
Yeah.
But then if I'm going to say the question is, did a dinosaur evolve into birds?
Right.
Right. That one, I don't see how you can repeatedly address that question.
Right. Right.
That's different. And yes, you can prospectively take some sand and gravel in an aquarium and apply some waves to it and show how its sediment forms, you know, and that may explain some geological process. And you did that prospectively. And to the extent that you don't have lots of assumptions about what happened in your tank versus what happened in Mexico or whatever it is,
Yeah.
you're studying, you know, there's some validity to all that. But you always have to be careful about the assumptions you're making and the question you're trying to answer.
Okay. Thanks.
Ken Wolgamuth, go ahead.
I've listened to John Sanford talk about his view about genomes degrading. I think that's the wording. And I've read his book also. It made it sound as though that the human race would disappear before too long in the way he described it, the way I heard it. What was I missing? And because, in fact, our population is growing larger.
Well, the population may be growing larger, but that does not mean that the genome is becoming more robust over time. I think John Sanford's point is that the genome is becoming less robust over time, that we're becoming weaker, if you will. And I mean, his main point is if you take 100 mutations, like each generation of humans, the next generation has something like 50 mutations more than the previous generation. And we also know that the large majority of mutations are detrimental. So we're accumulating detrimental over time. That doesn't mean we don't have a larger population than last year. It just means that we're getting weaker over time. That's his thesis.
Sounds baffling, but I'm not a biologist, so I'll leave that. Thank you.
Lothar, did you have a question? Lothar?
Yes, I did. You may be familiar with Stephen Meyer. What is your opinion of intelligent design?
Oh, I'm a big fan of intelligent design and a big fan of Steve Meyer. I guess you're talking about capital I, capital D, intelligent design, right?
Right. Okay, Wikipedia calls it a pseudoscience, but anyway.
Yeah, well, I certainly don't appreciate what Wikipedia says about that. But I think there's quite a lot of scientific evidence to show in favor of intelligent design. Like we all know that whenever you read a book, whenever you see a pile of information like that or computer code, you know it came from a mind.
Mhm.
And we know that the living things contain more information than almost any book you've ever picked up, and so we all know that that kind of stuff comes from a mind, and that's their argument. And I believe it.
Randy Lewening, did you have a question?
I did, yeah. No, thanks. This is a great discussion. And Rob, one thing that's sort of jumped to my mind as we're going through this is, in a lot of the experiments that you described, they had with E. coli, 20, 30,000, 40,000 generations. One of the interesting things is when you look at that number of generations and apply it to a mammal, like a human being, you get into the 3, 4, 500,000 years. And when Hugh Ross kind of looks at the history of Earth and climate change, we have ice ages about every 100,000 years. So if rice has a problem going from a warm tropical environment to a less tropical environment, we might have it's a big change in environment where you go from sort of the current environment to an ice age. So that's if the genetic changes, even if they were beneficial, are happening at that slow space, it's more viable for E. coli, but problematic for other creatures.
Exactly, unless the creatures have built-in adaptability mechanisms, right? Right, which is really that's how we're made. We're made very robust. Good point.
John Yu. Go ahead, John.
Yes, hi. Hi, thank you. So listening, so far the idea came to me, in fact, the human race is getting weaker physically, as you said, right? Through all these mutations, we've got more mutated genes, there's more cancer prevalent, so on and so forth. But also, if you look at the biblical history.
Oh, you muted yourself, John.
Sorry. So if you look at the biblical history, humans used to live 1,000 years for a long time. And then all of a sudden, we are now living only 100 years, 120 maximum. And so I'm sure there's some biological explanation for that. We can find that. And that would be a very interesting study if you can find it. And because this sort of agrees with your statement that we are getting weaker physically, not mentally, but physically. Okay. And so that's one thought, it's a comment. But it would be a good area for people to look into. What is it, that limits, from a scientific point of view, that limits the human life? And if they can do some research, come out with some kind of data that direction, that would be interesting.
Yeah.
And the other one is that the person's name I wanted to think about is Gary Habermas. Okay, so he's got these 13 evidences that most atheist agnostics would agree, and he has certain criteria, right? Certain evidences that the Bible claims about the resurrection of Jesus Christ. And then, according to him, he goes through the argument, these are all high confidence evidence. Okay, but I would like to maybe, suggest that using your thinking, along that line, come out with a parallel type of approach that can
It's called the
complement.
minimal. That's called the minimal facts approach, I believe.
Right. Okay, minimal fact. Yeah.
Yeah, making an
I don't know
argument
if-
based on what the academic consensus can agree upon.
Right.
Right. They can agree upon, but it's a high confidence. So that's a new thing I think, Rob, you've introduced, from a logic point of view or from some point of view, right?
Yeah, thanks for your comment. Some people believe it's very simple belief that the length of the telomeres in your DNA determine how long you're going to live. They get shorter over time. Every time you replicate cells, the telomere gets a little shorter. So you only have so many replications in your life, and then you're out of gas. So some people think if you can just artificially lengthen your telomeres, you could live forever. But I think that's way oversimplified.
Does that mean that people in ancient times had longer telomeres?
Yeah, I mean that's a hypothesis. I would think that's
has shortened the telomeres.
Yeah, God could have just snipped your telomeres.
Whatever.
Yes. But I'm certain that that's way oversimplified, but some people think that way. There's transhumanists out there that think that we're going to engineer better humans and we'll live forever, and that's their first approach is to lengthen your telomeres.
I see. Okay.
Okay, I've been dying to add my own question, if you don't mind, if nobody minds.
Sure.
Mine is really kind of going the other way and saying, okay, well, let's see if intelligent design can do something that, or I should say, if evolution can do something really remarkable. And so the thing that I'm thinking, this is only one example of things that I'm thinking of, but can evolution produce, let's say, the process of glycolysis, which is one of the steps in metabolism, and it's the one where you take sucrose, the simple sugar sucrose, and you create from sucrose pyruvate. So sucrose is I should say glucose. I should have said glucose, not sucrose, but glucose is a 6-carbon sugar, and pyruvate is a 3-carbon sugar. Well, that's an essential process in order to get to the citric acid cycle and the electron transport chain and the creation of ATP. Okay, so how good is evolution in creating the process of glycolysis, which is a 10-step process, all of which are required, as I can see it, in order to make pyruvate. And each step requires a separate enzyme. And each enzyme is a separate protein, which is generally hundreds of amino acids or even thousands of amino acids long.
Right. And if you only had one of those, where would you get? Nothing.
You don't, well, yeah, right. I mean, you don't have anything. So, and that's 10 steps. That's how has anybody ever suggested an evolutionary process that would get you glycolysis? I've never heard of it.
I haven't heard of it, but I know
but then are very... let's not stop there, because let's take, for example, the Stanford medicine map for metabolic processes, which has glycolysis on it, as well as the citric acid cycle, as well as lots and lots of other steps. And basically, I counted the number of enzymes just on that chart alone. It's 256 or 254 enzymes. And that's not really all we need. I mean, that's just the metabolic processes in human beings. This really seems to me like magic. All of these enzymes that were created somehow, I don't see that evolution has a prayer of creating even one of them, much less 256. It's magic as far as
you make a very good point. I really appreciate that. And I think it's especially powerful when you point out circular causality, meaning the chicken and egg type phenomenon that are all over the place in life. You can't have A without B, but you can't have B without A.
Mm-hmm.
And how does an evolutionary process overcome something like that? Good point.
Anyone else? I didn't mean to shut everybody up, but I did want to get my point out. But
So are the time scales? I'm an engineer, so most of this stuff is above my head. But the basic question is, I mean, the time scales of evolution. Is this not one of the big Achilles heels of that? There just hasn't been enough time for all this innovation, right? Since when life first started. Is this not one of the sort of the more fundamental problems? I don't know. Is that
Yes. Yeah, I agree. It's often called the waiting time problem. And I'm thinking of a paper that came out where they focused on whales and the supposed sequence of evolution of whales. And they looked at how many things would have to be majorly changed in order to get from a land animal to a whale. And the evolution story says that all happened in like 10 million years. But the waiting time problem, of making hundreds of modifications, it just doesn't happen in the time that's allotted. So it's a good argument.
Rob, would you mind closing us with prayer?
Sure.
All right.
Very much. Lord, thanks so much for this opportunity to be here. Thanks for gathering together these people, Lord. Lord, we give you the glory for all that you've done, for all that you've created. We appreciate your genius and we marvel at it. We worship you for that, Lord. And Lord, we pray that you would equip us as we go and talk to friends and coworkers and skeptics, and give us the wisdom, the right words, so that we can give you the glory and point to you and help to overcome what Romans chapter 1 says, where some people will not see all that you've accomplished, even though it's obvious. Help us, God, to help people open their eyes and make your name famous. And again, we thank you for this time and pray your blessings on everyone gathered here for their weekend ahead. Pray in Jesus' name. Amen.
Amen.
Thank you, Rob.
And Rob, thank you so much.
Thank you very much.
This is been wonderful, I think, and I really appreciate your spending time with us and sharing with us your thoughts on these very important issues.
Yeah, thank you, Kirby. And if you can get me connected to Fuzz, we can do more of this.
Yeah, I would love to. Unfortunately I don't carry much weight with him, so all right, I'm going to stop this recording though.